Hi, welcome to the verbal explanation on topic 7, which is on the aspect of plant and animal physiology basic procedure. We will begin discussing this topic on plant physiology procedure, and eventually we will move on to the aspect of animal physiology procedure. For plant physiology procedure, we will focus on the separation of plant pigments using paper chromatography method. Paper chromatography is a laboratory method that is used to separate plant pigments from plant based on differences of solubility of the plant pigments. Chlorophylls are the pigments that is green in color that is present in the chloroplast. Specifically, chlorophyll could be found in abundance in the thylakoid of a chloroplast. This is a typical experimental setup of paper chromatography to separate different chlorophyll pigments. The details regarding the procedure on the experimental setup is described in the laboratory manual on Lab 7, which is doing experiment on plant physiology. This is the expected outcome of a typical paper chromatography in separating chlorophyll pigments. If you have carried out the separation successfully, you will observe 4 different chlorophyll that differs in terms of color. The first type of chlorophyll that is nearest to the center of origin is greenish yellow in color. This chlorophyll or this color is identified as chlorophyll A. The second type of chlorophyll that will be observed is called chlorophyll A. Chlorophyll A is colored as in blue-green color. The third type of pigment that is distantly away from the point of origin, which is visible to us as in yellow color, is called or is identified as the xanthophyll. And the last type of chlorophyll that is furthest away from the point of origin, that is visible to us as in yellow-orange color, is identified as the keratin. After that, you could actually determine the RF value. RF value or the relative distance value could be calculated by this formula, which states that RF value is equals to distance that is traveled by the compound over the distance traveled by the solvent. We try to calculate the RF value of chlorophyll B. The distance traveled by the compound that is indicated by the example shown here is 2 cm. And the distance traveled by the solvent that is indicated in this example is 12 cm. So in order to get the RF value, you need to divide 2 over 12. High RF value, as being shown by the keratin, indicates that this substance is very non-polar. Low RF value, as indicated by chlorophyll B, indicates that the substance is very polar. As what is being shown in this table, RF value usually will range from 0 to 1.